Welcome to the fourth and final lesson on the immune system. Before we start, I'd like to explain how this lesson will work today. Throughout this video, I will ask you to pause and carry out a task on the Google document. So, today's lesson is about HIV and viruses. I'd like you to pause the video in a second and answer the following questions. What are the differences between HIV and AIDS? And why do people use them interchangeably? Please complete this using the link on the Google document in the introduction section. HIV stands for Human Immunodeficiency Virus. This means that it is a virus that affects the human immune system. HIV eventually leads to acquired immune deficiency syndrome, AIDS. AIDS is a condition where the immune system deteriorates and eventually fails. This makes someone with AIDS much more vulnerable to other infections. We'll look at this in more detail later. HIV infects helper T cells. Because these are the cells that are infected by the virus, we call them host cells. Remember that helper T cells activate phagocytes, cytotoxic T cells, and B cells, so they're hugely important in the immune response. Without enough helper T cells, the immune system is unable to act effectively. People with HIV develop AIDS when their helper T cell count drops too low. HIV has a spherical structure. It has a core, illustrated in the dark yellow part of this diagram, which contains genetic material, RNA, and some proteins, including reverse transcriptase. The core is surrounded by a protein coat called a capsid. There is also an extra outer layer, which is a lipid membrane. Sticking out from the lipid membrane are lots of copies of attachment proteins. These help HIV attach to the host helper T cell. Pause the video now to complete task 1 on the Google document. HIV and all other viruses can only reproduce inside host cells. This is because they don't have the equipment, including certain enzymes and ribosomes, to replicate on their own. Here's how HIV replicates. First, the attachment protein attaches to a receptor molecule on the cell membrane of the helper T cell. This allows the capsid to enter the cell, where it uncoats and releases the RNA into the cell's cytoplasm. Inside the cell, reverse transcriptase makes a complementary strand of DNA from a viral RNA. From this, double-stranded DNA is produced. Then comes the nifty part. The double-stranded DNA is inserted into the human DNA, so that host cells start making more viruses, which can infect other cells. I understand that was a lot of information. So here's a video to go through it again.
HIV targets CD4 plus cells, meaning cells that have this specific molecule called CD4 on their membrane. Macrophages, T helper cells, and dendritic cells are all involved in the immune response and all have CD4 molecules. Therefore, they can be targeted by HIV. The CD4 molecule helps these cells attach to and communicate with other immune cells, which is particularly important when the cells are launching attacks against foreign pathogens. So this little molecule is pretty important for our immune system, but it's also extremely important for HIV. HIV targets and attaches to the CD4 molecule via a protein called GP120, found on its envelope. It gains access into the cell. HIV is a single-stranded, positive-sense, enveloped RNA retrovirus, meaning that it injects its single strand of RNA into the T helper cell. The retro part of the retrovirus isn't referring to its style, but refers to it needing to use an enzyme called reverse transcriptase to transcribe a complementary double-stranded piece of proviral DNA.
Proviral just means that it's ready to be integrated into the host's DNA. So it enters the T helper cell's nucleus and pops itself into the cell's DNA.
Pause the video now and have a go at task 2 on the Google document. People with HIV develop AIDS when symptoms of a failing immune system appear or their helper T cell count drops below a certain level. The initial symptoms include minor infections, such as infections of mucous membranes. These include the inside of the nose, the ears, and the genitals. As AIDS progresses, more serious infections occur, such as diarrhea and bacterial infections. In the late stages of AIDS, there is a very low number of immune cells. This means that the patient is prone to very serious infections, such as parasite infections like toxoplasmosis, or infections of the respiratory system, such as candidiasis. Antibiotics don't work against viruses. They only kill bacteria because they target specific bacterial enzymes. There is no cure or vaccine for HIV, but there are ways to control it. Antiviral drugs can be used which target virus-specific enzymes, such as the reverse transcriptase in HIV. However, the best way to control HIV is to reduce its spread. HIV can be spread via unprotected sexual intercourse and infected bodily fluids, such as blood from sharing contaminated needles. Pause the video now and have a go at task 3 on the Google document. For homework, I would like you to answer the exam-style questions on the Google document. If you're really interested in this topic or want to challenge yourself further, you can have a go at the extension task.